So it is like last week of the term. So thank you for everybody who's been coming to the seminar, making it great. I will say we are still soliciting nominations for fall and winter. We have some, and the committee and myself are going to be going through those and trying to start getting back to people who submitted nominations about inviting. But if you have ideas, the link was out there. I might try to send it out again before the end of the year. But please submit nominations, especially, we want to really capture the breadth of stuff that people are interested in ecology and evolution. So, so please do submit nominations. Secondly, I think because it's the end of the year, I also just wanted to acknowledge, like Peter, there are a lot of other students who've helped out with this committee. So I'm super helpful or super thankful to them. Also, because this is like end of the year, there've been a lot of things going on. We have a lot of people like graduated. A lot of people did proposal exams this year. A lot of people had like their DAC meetings and faculty are teaching. So whatever, congratulations, everybody. And partly for that, there's still going to be tea time. I wish I brought popsicles because obviously it's like 80 degrees in here. So there will be food. There's nothing super ice cold, but there will be tea time after the seminar hosted by me. All right, so with that, I'll get on. As I said in my email, I'm going to try and keep this shorter than a normal full-length seminar so we can all, don't get heat stroke, and also just so we can go hang out, enjoy the day. So I'm going to be talking about a project that uses some methods that we've used for several years in my lab, but it's not something that I think I've talked about here or in any of our various journal clubs or other communities. So I'm really excited and happy to talk to you about that. Other people I want to acknowledge, especially the folks in my lab. This is a more recent picture where we finally got all of our lab t-shirts on together. This is an older one, but I still like it because when I was on sabbatical and I got to bring people out to Scotland to come visit me, which was lots of fun. The project I'm going to talk to you about, we also have some awesome collaborators, Victor Torres, who's now at St. Jude's, Brian Geisbrecht at Kansas State, and Terry Medler in particular, who's up at Providence in Portland, and some other people for other projects. All right, so if you don't know, and if you only know me as like the guy who sets up tech at the seminar and you don't know any science that we do, I nowadays describe what we're interested in as evolution of host pathogen systems. So two of the big questions we're interested in the lab are how have hosts, particularly animals, including humans, evolved to defend themselves against pathogens. In particular, in our lab, we're interested in pathogenic bacteria, which I'll tell you a little bit about. And then conversely, we've been starting to look at a little bit more in the last several years about how microbes themselves actually evolve and adapt to different challenges they encounter in host environments. This could be, immune cells that are trying to eat them, like this one here. This could be other microbes. Now I have a student working on viruses, bacterial viruses in the lab, which is really fun. But I'm just going to tell you about one story, which is sort of more on this, addressing this first question here. And so as my talk also alluded to, this is getting into an area that I was not really interested in or thinking about when I came to UO almost 8 years ago, but we've gotten really interested in the last several years, which is the skin. So the skin does a lot of stuff for us. So it's the largest organ in the human body. It's important for preventing fluid loss, temperature regulation, protection from a lot of chemical and environmental damage like UV rays. But importantly, it's also both a habitat and a barrier against microbes, right? So there are microbes that are residents of human skin in some of these different micro niches, either on the surface, within hair follicles and sebaceous glands, things like that. It's not as numerous or as diverse as things like the gut microbiome of animals, but there are definitely sort of keystone taxa that we think of as being residents of skin. And, so think about that question that I posed a slide ago about how animals defend themselves against pathogens. We've thought about this in a lot of different contexts. I'll give you some examples of that. And many people in the field have thought about this in the context of sort of traditional immunity factors, things like antibodies and the adaptive immune system. But in sort of going into this project, what I was really interested in was how has the skin itself as a barrier potentially evolved in response to pathogens, and how has that evolution shaped susceptibility to certain microbial pathogens. So this was kind of the big question going in. And pretty much all the work I'm going to show you today was done by one amazing senior research scientist in my lab, Kristen Kohler, who's in the back there. So if I mess anything up, I might direct questions to Kristen. And I thank you to Kristen for letting me present her work here today. Okay, so in trying to understand how host genes or proteins may have been shaped by pathogens. I'm going to introduce a method that some people might be familiar with, others maybe not, that we've used a lot in my lab to think about, to try and identify instances where we see repeated natural selection, particularly looking at positive selection in protein coding genes. And this is through calculation of a metric called DNDS, also sometimes referred to as omega. And so the idea of DNDS is that we're, for a single or maybe many protein coding genes, calculating a ratio, which is the rate of coding or non-synonymous substitutions versus the rate of silent or synonymous substitutions. And so the way, I think I had this in my tenure talk from several years ago, analogy that if you look at a single codon, a single protein coding gene, what we tend to see in most organisms is this accumulation of silent or synonymous substitutions that don't change the underlying coding amino acid. And this, when you look at this over long evolutionary timescales, this often reflects the force of purifying selection, right? That mutations that alter amino acid state tend to be deleterious more than they are beneficial. And so for many protein coding genes, if you calculated DNDS across codons of many genes, what you tend to see is this very low DNDS, an enrichment for silent substitutions relative to coding substitutions. But in considering how pathogens may have evolved or even co-evolved with hosts and driven repeated cycles of positive selection, what we're often interested in is looking for instances where a codon or a gene is evolving more rapidly in some ways than we would expect due to chance. So it's accumulating amino acid substitutions more rapidly than certainly would be explained by purifying selection, and even more rapidly than would be expected just by random genetic drift. Okay. And I think another key here, and this, I'm not going to belabor the point too much about like the benefits and drawbacks of DNDS. There's a lot of ways to look at positive selection, natural selection within populations versus between species. Where I think this method has been really useful for us is particularly looking at comparative differences between species and also instances where we suspect there hasn't been sort of like one selective sweep going on at one site, but where you have genes that are subject to repeated episodes of selection, often in response to some other evolving antagonist, like a pathogen. Okay. And I think I just explained some of the underlying logic here that, in instances where you have two interacting partners, say this is a protein from a pathogen that's interacting with a host protein and contributing to disease in some way, that the forces of positive selection will drive the spread of mutations that sort of interfere with that interaction. Of course, then we have pathogens, whether it's single species or other species in the system, that can then evolve to reestablish that interaction. And this is also is explained in part by theories going back all the way to the 70s with Lee Van Dalen and his Red Queen hypothesis, considering explanations for why everything from why sex is beneficial in plants and animals to forces that might drive coevolution in antagonists in nature. Okay. Okay, so we're not doing this between Disney characters, but in our case now, getting back to the skin, what we started to do was look at evidence of repeated positive selection, right? That kind of potential arms race-like dynamic happening in genes that are sort of structural or the core components of skin, generally conserved in all mammals, but we were focusing primarily on simian primates. So this includes the apes or hominoids, old world primates, and new world monkeys. Okay, and again, just for context, this is not the first time that some of you might have heard me talk about this approach or this methodology, and other people in the field have used this to look at different types of interactions. So, before I came to UO, we saw a sort of scenario where evidence of positive selection in a gene called transferrin, this is an iron transporter present basically in all animals, is rapidly evolving in a region that we later showed interacts with a bacterial receptor that targets transferrin as the nutrient iron source. So this was evidence that we then followed up with experiments to show, to suggest that natural selection in transferrin has actually been driven by interactions with these nutrient acquisition receptors in bacteria. And then more recently in my own lab, we've again used this type of approach to look at cell surface proteins that actually serve as like anchor points for microbes that colonize different host surfaces. So this was done by Emily Baker, a former postdoc in my lab who just headed off to Europe, where she showed that we see evidence of repeated positive selection in many sites in a gene called CCAM1, which is on many epithelial surfaces at regions that are known to interact with proteins called adhesins on bacteria that allow them to attach to those epithelial layers. And one thing I'll note here, in contrast to what I'm going to show you in a minute, is that in both of these cases, we had structural and genetic, and then also I'm not showing you experimental data to support the notion that the selection may have been driven by and certainly impacts interactions with pathogens. But we don't necessarily, everybody likes to have this, make this conclusion of like, oh, can you tell me like which pathogen drove selection at which point in time in history of certain organisms? We generally lack the power to do that. And partly it's biology. So in this case, these transferrin receptors, these TBPs, are present in many different bacterial genera. So they're not sort of restricted to one group. Similarly, there are many different unrelated adhesins that through convergent evolution target the same host protein. And so, as a biologist, I can always tell people like, we don't really have a lot of resolution on what was driving this kind of interaction. This story today, I think we have a little bit more. There's still a lot of questions and there's some hand waving, but I'll tell you a little bit more about that. Okay, so now think about skin. So I didn't know a lot about skin before we started getting into this, but it turns out the skin is not just a simple layer, right? We have regions, a stratum corneum, and this is again focusing primarily in terrestrial animals like humans, the sort of called corneal layer of the skin, which are mostly dead keratinocytes, this layer called the stratum granulosum, and then the sort of lower layers, the spinosum and basal layers. And an important component of the skin is this little structure that I've enlarged here called a desmosome. So if you ever took a cell biology course, you might have come across this before, but a desmosome is essentially a large proteinaceous structure that forms on two adjacent cells. And Kristen's analogy for this that I really liked is that it's basically like rivets, like really tight connections that anchor two cells together. It's then anchored to intermediate filaments on either side of the cell, but there are extracellular proteins that form that connection between the two cells in the desmosome. And so these are really important in many tissues, not just skin, but skin-specific desmosomes are what basically give us a watertight and other, layer against a lot of, invasions, whatever. Okay, moving on. So some of these different extracellular proteins, there's different families. One of the major ones that I'm showing here are called desmogleins or desmoglines. So there's 4 in primates and humans. One of which, so desmoglein 1 is sort of enriched in the outer layers of skin. Desmoglein 3 is enriched in the lower levels. Desmoglein 2 and 4 are primarily expressed in other tissues like the heart, for example. And so in doing this phylogenetic analysis, what Kristen and I found was that there was really strong evidence of repeated positive selection of elevated DNDS in desmoglein 1, but something that we didn't see in any of the other desmogleins. So what was going on there? What was cool was when we then looked at like what is the region, what are the sites in this protein that are giving us that signature of repeated positive selection. It turned out there were only 6 sites, and they're all in this region called ectodomain 3 or EC domain 3. So this is the extracellular part of the desmoglein. This is the transmembrane domain, and then what would be in the cytoplasmic part of that cell. And so out here is where it's contacting other desmogleins to form that junction. But all of our rapidly evolving sites were clustered in this one domain, and they were separated. So there's 5 here that were together, and 1 that was about 100 amino acids apart. But when you look at them in a 3D reconstruction that Kristen generated, it turns out all 6 of those rapidly evolving sites in primates cluster into one surface, right? Okay, so separated a little bit in space, but structurally all on one surface outside part of the protein, okay? Functionally, this doesn't really do anything that we know of for the host, right? It's not directly involved in desmoglein-desmoglein interactions. There's no other sort of thing that's binding to this. So then that's the question, is why in this really conserved structural skin protein do we have very strong signs of repeated positive selection, but it's restricted to 6 sites in the surface that we don't really know what it does. And so I'm going to cut out a lot of research and questions and everything else to tell you that in a second. But in order to provide context, I have to introduce a microbe into the story, which are staphylococci. So staphylococci are part of the group of gram-positive bacteria. So they have membrane, large, thick outer cell wall. It's one of the most abundant genera present on animal skin and also particularly in the nares, so like your nostrils. And the one that most people have probably heard about is this one, Staphylococcus aureus. So Staph aureus is present primarily in the nose or some other skin regions of about 30% of people in the human population. It's thought of as being fairly human adapted, or we were likely the reservoir for this organism, although it can cause infections in other animals as well. In humans, it's the major cause of skin and soft tissue infections, but it's also a leading cause of lethal bloodstream infections, hospital-associated pneumonia, osteomyelitis, so like bone infections. It's a really nasty bug. And I'll get back to this point about toxins in a second. I just added this really quickly because I think for this audience that's not sort of necessarily infectious disease focused, I wanted to drive this home. That there recently, I think until recently, there wasn't a lot of good estimates about how different bacterial pathogens were contributing to disease in humans. And there's this series of studies, this is from one of them, that took data. It was the first attempt that I'm aware of to really get a good estimate of global deaths from infectious disease globally across all countries in 1 year. This is data from 2019. And I should say it's a little misleading because they're excluding tuberculosis here. So Mycobacterium tuberculosis, there's a lot of good surveillance and we have good estimates on disease levels there. Omitting the years of the COVID pandemic, TB was the leading cause of infectious disease related death in humans on the order of like 1.5 million people a year. But from this and related studies, what became really clear, this is Staph aureus right here, and this is numbers of deaths categorized into different types of diseases. Don't worry about the colors. But based on this and some other studies, the estimate was that Staph aureus was causing over 1 million deaths a year in 2019. It was second only to tuberculosis. So that's more than malaria. That's more than HIV. So it's a lot. And it's sort of spread against both middle, low, and high income countries, right? So we still have a problem with Staph aureus infections in the United States and Western Europe, Canada. In addition, so methicillin-resistant Staph aureus, or MRSA, which is another term some people might have heard of. So this is one sort of group of antibiotic-resistant Staph aureus from related studies is one of the leading causes of antibiotic-resistant infections in many years, including 2019. As I said, they cause infections in a lot of animals, particularly in dairy-producing cattle. Actually, it's one of the leading causes of bovine mastitis. So infections that dairy cattle get that are a big economic burden on the dairy industry. And then, as I said, part of the reason that staph, it's a resident in humans, but it can infect other animals. It's in your nose, but it can cause bone infections and bloodstream infections and pneumonia. And part of the reason we think it's able to do this thing, have these different phenotypes, is that it encodes what we call a lot of virulence factors. So these can include things to shut off the immune system, but they can also include toxins that destroy or disrupt host cells of tissues in different environments. Okay, so among these, there are many well-studied toxins and staph. Many of them kill immune cells or things called superantigens that just lead to an over-inflammatory response. These are things like the toxic shock syndrome toxin that was related to tampon use back in, I think, the 80s for the most part. One of the lesser known toxins, the less studied toxins in staphylococci are called exfoliative toxins. And they get their name because of this phenotypes. This is a cross-section of human skin that's been from a patient that was infected with this toxin-producing Staph aureus. And you can see that that's sort of, here's the corneal layer of the skin up here. Here's the sort of granulose, the lower layers down here. And this is basically a blister, right? So we've got space where toxin has led to denudation, sort of removal of the outer layer of skin. So this toxin, through a lot of cool work that I'm not going to go into, has been linked to a couple interesting diseases in humans. So staphylococcal scalded skin syndrome. So this is primarily infects newborns and infants. Bullous impetigo, so this is a localized blistering disease. And then particularly in things like farmed pigs can get a disease called exudative dermatitis, which is similar, where they have basically blistering over large parts of their body. Really horrible infection. This is the most detailed picture I'm going to show you related to that. Don't worry. And so how does it do this? What was found a couple of years ago, several years ago now, is that the exfoliative toxin is actually a protease. Okay, so it's not something like a pore-forming toxin that are other ones that staph produces, but it's a protease, and it's a highly selective protease. The only substrate that's known for this protease to date is desmoglein 1. So it cleaves desmoglein 1. Where's my mouse? I lost it. In these extracellular domains, it leads to a release, and now you've lost that rivet, that desmosome formation in the skin. And selectively in the outer layers of skin where desmoglein 1 is present and not desmoglein 3. Okay. I'm going to actually come back to this and sort of skip over it in the interest of time, but the short version is that these aren't just in humans. There are other staphylococci that infect other animals that encode homologs of this toxin. We can talk about that later if there's time. Okay. So, all right. So coming back to evolution, desmoglein 1, this surface here that's rapidly evolving. Somebody is calling me. No, I can't take that right now. We don't actually, like these other studies I've done, we don't have a co-crystal structure of this toxin in complex with desmoglein 1, but we do know it only cuts in one site and it's conserved in all the different related exfoliative toxins. And we know where that site is, and it's right here. So we have, now I'm just honing in on just these two domains of desmoglein 1. You have this one surface, 6 amino acids right here, and about a few amino acids away from this most C-terminal site is the known conserved cleavage site for staphylococcal exfoliative toxins, basically right next door. Okay, and just to sort of show this in the sequence, here are those 5 clustered rapidly evolving sites in the sort of other portion of the third ectodomain. I'm just false coloring amino acids that differ from humans. So a lot of the variation we're seeing relative to human desmoglein 1 is present in old world primates and also in new world primates, although we have fewer of those. And then here again, sorry, is the cleavage site. So it cleaves between the E and G residues here. Okay. So again, so based on like prior work by us and other people, we would say, okay, maybe this is an explanation. Maybe this is variation in desmoglein 1 that could provide protection against this bacterial toxin, okay, without knowing anything about the structure necessarily of how it targets it. Right, so that was sort of our next question, is how does variation in this toxin affect exfoliative toxin tropism? And this is like where a lot of studies in my lab goes. We use the genetics to generate hypotheses, and then we try to test those in the lab, often using molecular approaches. So in this very straightforward schematic, what we can do, and this has been used in this field before, is express recombinant desmoglein 1 that just has those 4 ectodomains fused to GFP. So if you express this in cells, they secrete it into the media, and then you can collect it. I'm explaining it like it's really straightforward, although Kristen knows that getting cells to behave and make proteins you want is often a very frustrating endeavor. But we can take this purified desmoglein 1 that's recombinant, so we can make this from different species. We can introduce mutations. We have a lot of control. Incubate that with purified exfoliative toxin that we can actually purify from staph or express it in E. coli. And then look on using a Western blot and a GFP-specific antibody how this substrate is cleaved through time. So if there's no cleavage, you sort of see a constant band as you move across taking samples through time. But if you get cleavage at that particular site, what we expect to see is this gradual reduction in the band. So now we have this cleaved fragment here, which is indicating activity of the toxin. All right, so that's how the assay kind of works. This is what it looks like in practice. So here's human desmoglein 1 incubated with one variant of this toxin called exfoliative toxin A. The time is across the top in minutes. So within a few minutes of incubation with exfoliative toxin A, human desmoglein 1 is very rapidly cleaved and pretty much completely by about an hour under these conditions. Okay. And I should also say, for context, that this activity is known about in humans. You can actually model staphylococcal scalded skin syndrome in mice, because laboratory mice are also susceptible to this toxin. Okay, but beyond that, and I should say it was known that other farm animals, cattle, pigs, more distantly related, are not susceptible. But susceptibility to this toxin in primates, to our knowledge, had never been previously investigated. So all of this was new to us. Okay, so we see in some instances, like gorillas appear to be susceptible just like humans, orangutans, right? These are all apes that are fairly closely related to humans. But in contrast, a lot of non-human primates, so these are rhesus macaques, night monkeys, a New World monkey, baboons, we sometimes see some low level of activity, appear to be entirely or partially resistant to the toxin. Okay? So even though they're more closely related to us than laboratory mice, from this assay, it seems like some of these primates are actually resistant to exfoliative toxin. We can look at this data different ways. You can plot, the band density of the cleavage fragment and try to quantify cleavage across different species. We see similar things. This is using a different exfoliative toxin variant called exfoliative toxin B. And we see similar trends. For a lot of the rest of the talk, I'm going to show this data as basically a heat map. So the idea is that the warmer colors indicate more complete cleavage at across different time points, same as before, 0 to 120 minutes. And now we're going to look at this data in humans, other species, for the exfoliative toxin A and the exfoliative toxin B variants. And this is what it looks like. Okay, so roughly grouping by phylogeny here, we can see that a lot of apes, again, are susceptible to both exfoliative toxin A and B. Many old world monkeys appear to be resistant. So rhesus and baboon, we see partial resistant, full resistance basically in rhesus. New World monkeys, many of them seem to be resistant. And then we recapitulated what's been seen before in the literature, that mice are susceptible to both of these toxin variants, and pigs seem to have some susceptibility to ETB, but not ETA. So all of that piece was already known. All right. So, okay, so based on what we had seen from the phylogenetics, that there's some evidence of selection that could shape this variation in non-human primates. This was all mapping up. But let's see. So one thing, I just kind of added this really quickly because it's experiments that we've been trying to do for a really long time, and Kristen's finally starting to get some results on this with our collaborator up at Providence, Terry Medler. But what we wanted to do is, in addition to looking at this in vitro assay, which in the literature all matches up with what people see in vivo, is to actually test, is the primate skin actually susceptible to this toxin? So you can do an experiment kind of showing what I showed earlier, where in this case, this is a control of just tris buffered saline, where you have an intact skin layer. And this is, I should say, using human skin samples. You can treat with, in this case, 100 micromolar ETA and see the formation of the blister in the actual tissue. So we've been doing these experiments. I think it's too early to say conclusively. We were initially seeing a lot of stuff that's consistent with what we expected in vitro. But experiments are still ongoing to see if this also pans out in an ex vivo setting. So stay tuned for that. Okay. So the other question at this point would be, are those sites that we identified, are those rapidly evolving positions actually sufficient individually or in combination to affect this phenotype? And so Kristen did a lot of mutagenesis, focusing on these rapidly evolving positions. I'm going to show just one for which she's got a lot of data, which is position 271 right here, which is a Q in humans, again, relative to the cleavage site. So what initially Kristen saw was that when she takes humans and introduces just a single amino acid change, in this case to resemble the position at different non-human primates. So I think this is what's present in some old world monkeys, and this is in some new world monkeys. That single mutation, interestingly, is sufficient to give you resistance to ETA, and reduces susceptibility to ETB, but not entirely. So this is also getting at, there's likely some difference, genetic differences in the toxins and different strains that are carried that affects the host species tropism here, which I think is pretty cool. But other mutations, so this is actually a variant that's present in human populations at reasonably high frequencies, doesn't, in this case, seem to have any effect. Or I should say low frequency, that one. We did this in some other systems too. So you can take baboon, which if you recall, is mostly but not entirely resistant to exfoliative toxin A. And if you make a single amino acid replacement at that same position to now resemble humans, that's sufficient to give you susceptibility that looks like susceptibility to human toxin, right? So we have both necessary and sufficient for this activity, which is great. I'm just showing some mutations at other positions. Capuchins, we tried to do the same thing to, enhance activity with a single amino acid change, couldn't do it. And same for night monkey. But yeah. So, at least in some cases, single rapidly evolving sites that show evidence of repeated positive selection are sufficient to be able to control either the loss or the gain or enhancement of susceptibility to this toxin. Cool. All right. So I am going to try and wrap up pretty soon. The last couple bits I'll mention are sort of getting at questions I typically get, which the first one being, okay, we talked about primates and Staph aureus, but you mentioned this toxin's present in other staphylococci. So what about susceptibility in other animals? We haven't done a lot with that yet. The one thing I'll tell you is that when you try to run similar phylogenetic analyses in other taxa, so this is Kristen looking at rodents, we also see some evidence of positive selection of high DNDS in rodent desmoglein 1. In this case, it was pretty cool. There's two positions that cluster in that same surface. One of them totally aligns with one of the rapidly evolving sites that we identified in primates. And there's a lot of sort of definitely coding variation in this region that we think could be important. Functionally, we haven't tested this yet. Could be an area for future work. Okay. Sort of similar in carnivores, although not as strong. And then, the sort of last bit that I'll show you is, again, we don't have a crystal structure. We don't know how this thing is interacting. Brian Geisbrecht is our collaborator at Kansas State, who's been trying to get a co-crystal structure of the toxin with the substrate and had a lot of difficulty with that. The most we've done is what everybody seems to do nowadays is like, let's just alpha fold it. So you can now try to take different proteins and see how they might interact using AlphaFold. Sorry, this is my movie. This is something that we generated. So those rapidly evolving sites are shown in blue in desmoglein 1. The cleavage site is in green. The sort of catalytic sites in the toxin are in orange. We're not super confident. Honestly, I'm just showing this because it's sort of fun and everybody always asks. We're not super confident that the models we're getting necessarily reflect the actual orientation, but we can definitely get models in which we see interaction. And so this might be sort of some kind of interaction that we're seeing that reflects how variation in this surface could impact then cleavage at the neighboring site. Okay, so I'll go ahead and wrap up. Quick summary. So we have now genetic evidence of repeated positive selection acting in this skin desmoglein 1. That's a component of the desmosomes in the skin in primates. The rapidly evolving surface of desmoglein 1 lies adjacent to the known cleavage site of a class of bacterial exfoliative toxins. And that variation of desmoglein 1, both among primate species and at single rapidly evolving sites, is sufficient to control this tropism. And then, a lot of outstanding questions we have at this point. Do we see evidence of selection acting on this gene in other taxa? Because we know the toxin is likely present in other species-specific staphylococci as well. We see evidence of that in rodents and maybe carnivores. We haven't done experimental work there. I think that'd be really cool to look at in the future. Can we sort of understand from the toxin side, like, can we actually trace maybe coevolution between a particular species toxin and the host? I think that would be cool. We haven't gotten to the point of actually trying to do that. And I think it would also require some knowledge of like, what are the actual regions that are interacting with this site between different toxins? And we don't have that information yet. I think you could sort of take a broader, I've thought about this, what this is telling us about disease ecology and the importance of these pathogens, not just in humans, which I talked about a lot at the beginning, but in other animals, right? So if we're seeing this much repeated selection, what that reflects is that, these things are acting, over millions of years, certainly in different extant or ancestral primate species, but potentially a lot of other species as well. And so, we don't know a lot aside from humans and farm animals about staphylococcal disease burden in other animals, but I think it's something that definitely we should be thinking about. And then I showed you again this one example of how this component of the skin has evolved in response to a bacterial toxin. We haven't really, this is an area that nobody's really sort of thought of before in this way, but it opens the question of like, how might other components of skin been subject to similar selective pressure in animals. So it's a big open question for us. All right. That's it for me. I want to again thank all the members of my lab, collaborators, funding. Thank you all for listening, for coming to the seminar, and happy to take any questions here or upstairs, possibly where it's cooler with food at tea time. All right, thanks. Normally, I'm the one running around with a microphone, but I think I'll stay here and just let you yell your questions if that's okay. Anybody? Rose? Yeah, so in case anybody got on Zoom. So I didn't show the, let's see, humans are right down here. If you look at this region, I'm not going to sort of compare it directly. They're very similar, right? And so I think if you want my guess, it could be essentially just more or less random, right? That maybe if there hasn't been a lot of selection in apes and there hadn't been in the sort of common ancestor and the rodent lineage that led to laboratory mice, then that could explain it, and that it's been more lineage specific. So maybe it's happening within certain groups of rodents, within primates. But yeah, I think there was an element of randomness here that if we just had checked in some other rodent, we would have said, oh, we can't model this disease here. And I should say that's definitely true for a lot of other staph toxins that we know the function of, is they don't work in mice. We do infections in mice, but, well, not we in my lab right now, but people model it, and that's a big issue, is that a lot of these toxins don't function in mice. It does in this case, but yeah. Anyway. Peter. This project from the start. The short version was that, we took existing data sets or data sets we had of like candidate genes in non-human primates that show that signature of high DNDS. And then what I did even sort of before roping Kristen into this was just like look at sort of cross-reference that with like known components in skin, right? And what's the overlap? You do see other interesting things. I mean, there are genes like filaggrin, which some people might have heard of because there's a lot of human diseases associated with mutations in that. It's another sort of structural skin gene. But yeah, that's the short answer. Maybe.
So when after the so one thing that's maybe interesting is so if you like zoom out and zoom out.
Yeah.
They're very different in the environment.
Yeah. So marine mammals would be a good one to look at. I don't know if anyone's seen if the expression, I think so one key would be like, where are these desmogleins expressed, in the skin in different animals? It's known in a lot of terrestrial mammals. I don't know if it's been looked at in, whales, dolphins. I think also, and then looking beyond that, I mean, we have these pathogens that we know infect avian species. I don't know for sure whether that sort of tissue expression is the same. I think that would be a key here, is like what our hypotheses would be. But yeah, the short answer is we don't know yet. Yeah, I would say in a lot of like aquatic or marine, non-mammalian species, the skin is also like, people like Brendan have told me it looks more like the inside than the outside almost, right? Like there's more mucus essentially on the skin and things like amphibians, and so how that affects these types of interactions might also be important. Yeah, Nelson.
You know how but you know how those like upstream of the beach site, how exactly they're affecting whether food is happening or not?
So these ones that, well, let's see. These ones here, essentially, right? Yeah. So we don't think they're affecting sort of normal function of the protein because it folds normally and sort of behaves normally as far as we can see. How they're affecting the toxin. Yeah, okay, so I didn't talk about that too much. Molecularly, what I think is happening is that, the toxin is, maybe a little bigger than one of these domains in terms of its like amino acid size. So it's probably docking. We think it's, and this is sort of true with other proteases, that they'll land somewhere and they might cleave adjacent wherever they went, right? So presumably there's a protein-protein surface that's required to recognize the target that's adjacent, and then it's cleaving there, right? That would be the general hypothesis. Like, and that might explain like why don't we see, at least in primates, why isn't there variation on the other side? And that just might reflect the orientation of where the toxin sits on there. Yeah. No, I'm looking at Kristen if that makes sense. I don't know. Yeah. Yeah. I think again, without like a higher resolution structural picture, that could drive, the hypotheses there, but that, yeah. Yeah, I thought about that at some point. We could definitely do that. We finally started doing ancestral reconstruction with other projects. I'm looking at T-Tosh in the back of the room. So I think the question there I had was, yeah, I mean, I guess how far back would be interesting to go. Like we could do the common primate ancestor for sure, and then test if there's susceptibility there. I mean, what would be great would be to reconstruct some ancestor of the toxin. So if we could take so I didn't get too much into this for the sake of time. There's a pig adapted staphylococcus called Staphylococcus that encodes homologs of this toxin. And then there's one that's known to infect or colonize dogs called Pseudointermedius. And they both encode sort of homologs of the toxin. So if we could do something like reconstruct, I think it would be cool. This is like my experiment. Reconstruct the, say mammalian ancestor and the common ancestor of the toxins that infected ancestral mammals. And could we see, start to look at coevolution in some ways from that standpoint? Yeah. But even on a shorter time scale, like apes or primates, we could do that. Yeah, we haven't done it yet. Right? Yes. Okay, thanks everybody.